Hi, my name is Michael Wells. I'm a senior graduate student in Guoping Fang's lab here at MIT, and I study autism. And the way I do that is through manipulation of mouse genome. I like to think about the mouse model as a way of reverse engineering the body. And so in particular, I'm reverse engineering the brain. So it'd be as if you, say, had a car and, like me, I know nothing about cars or how they work. And so if I was given an infinite amount of time and money, what I would probably do is remove one piece of the car at a time and then see if the car still runs or see if it makes this noise. So it's basically the same thing we're doing now. We're removing one piece at a time, just one piece of the genome at a time, and assessing the effects of the loss of that gene. The long-term goal of correcting those behaviors with either drugs or gene therapy or whatever magic thing we come up with in the next 20, 30 years. So I'm focusing on PATCH-D1 for my thesis because of a handful of papers that came out in early 2011. There are about 12 patients who are identified as having deletions or mutations of the PATCH-D1 gene, and these patients had autism and intellectual disability. Typically, when you're trying to study a new gene or a new protein that hasn't been studied before, one of the first things you need to do is figure out where it's expressed in the body. And in particular, I'm studying the brain, so I wanted to know where it's expressed in the brain. So in this image, I stained with an antibody directed against PATCH-D1. And I also stained with an antibody against GFAP. Now, GFAP stains glia cells. They're the supporting cells of the brain. And so what I was trying to do is see if GFAP and PATCH-D1 express in the same cells. And in this image, you can't really tell because it's a black and white image, but they do. So seeing this co-expression indicates that PATCH-D1 is expressed in glia cells. Now, why that's interesting is because there are very few known autism-related genes that are expressed in glia cells. There's a bit of a caveat to this, though. One PATCH-D1 antibody gave us this result. A different commercial antibody gave us an entirely different result, indicating that it's only expressed in neurons. So we're still in the process of ironing out which cell type actually expresses PATCH-D1, and that's one of the most important questions we have to answer. What made me choose this picture to submit it to the Koch Institute was the fact that it's only a single layer. So the confocal microscope can take multiple layers and you can stack those images, or you can look at a single plane. And so in this case, I only took a single plane. However, for whatever reason, it seems to have some sort of depth to it. And so that's what interested me the most about this, was it looked three-dimensional even though it was a single plane.